Welcome. This video is the final video in a three-part series. This will be a quick introduction to data management for new users of OMERO at the University of Arizona. Before we get started, this slide is a reminder that OMERO is only intended for the storage and management of de-identified data. That's not to say that we can't work with clinically relevant image data. We have some groups doing patient-related research who have image data that incorporates a unique identifier into the file name. The unique ID relates that image data to similar ID in REDCap, which is clinical data, or Open Specimen for biorepository type data. Those softwares are designed for this kind of data. The point being that it's not impossible to work with patient-related data. You do need to be very careful. Things to watch out for: protected health information of any kind is not allowed to be stored in OMERO, and this includes information that's embedded in the bitmapped image format, such as with medical imaging or slide label images, or in the image metadata, or that might be added by the user later with tags, key value pairs, and other annotations. There's a great deal of work being done by a number of different international groups to come up with a structure and methodology that's the best way of organizing the metadata that's associated with bioimaging experiments. Some vendors are on board with the process, and unfortunately, others aren't. And the details related to image metadata can get quite technical. In a perfect world, everyone would adhere to the highest metadata standards. But in the real world, in my experience, most labs could benefit from at least taking their current data organization up a notch or two. By being more methodical, labs will discover that their data is more findable, accessible, interoperable, and reproducible. These are the FAIR principles. Now, I'll admit that even though I've done a lot of reading over the last 2 or more years on data management, I would not consider myself a data management expert. And I hope that this presentation will give you some useful tips and tools to work with. Whenever you think about a project that needs organization, I like to come back to this quote from Stephen Covey, which is one of the 7 habits of highly successful people. And it says, begin with the end in mind. So what do you need from data to be able to reproduce it later on? In general, you want to plan to upload your data in the original file format. That's because OMERO uses the Bioformats software library, which allows it to interpret both the bitmapped image data as well as the associated metadata found in over 160 different proprietary image file formats. If you're not sure if your file format is supported, check the Bioformats list of supported formats. There'll be a link to the list found in the notes below this video. If needed, we can test upload a sample file to our OMERO training server to see if it's going to work. And unfortunately, some file formats don't work in their native version. If you discover that that's a problem that you're facing, please complain to your vendor, number one. And then if we do find a challenging file format, one possible option is to export the data, convert it to a different file format if we can. Converting the file to an OME TIFF would be our first recommendation. This format retains most of the instrument metadata, and it's recommended to convert to OME TIFF if you're doing a large area tile scan that isn't already stored in a pyramidal file format. Now, I may have mentioned in the other videos that pyramidal file formats are similar to what Google Earth uses. You start out with low resolution information, and as you zoom in, you get more and more detail up to the maximum magnification that's available. What you see on screen is the only part of the image that's loaded at any one time. This type of file format is much more efficient for the file server when you're working with really large data. The open microscopy environment is also working on a new file format called OME-ZAR, but this is still very much in the testing stages. TIFF would be the third option, but unfortunately there's a very real possibility of losing most of the instrument metadata, particularly if you have multidimensional data. So what is metadata? Computer experts would say that metadata is data about data. While that's true, we often need a more tailored definition for microscopy. And one definition might be all information that's needed to interpret, evaluate the quality, reproduce, and share microscopy images. If you think of the vast pool of image data information that your lab may have, metadata is both information about the unique piece of image data that you're looking for, but also how to find it using the information that the metadata provides you. Ideally, you have information about how the sample was prepped or the experiment was performed. You have image acquisition data, hardware configuration, your settings, your quality control, as well as your analysis and post-processing information. Without the appropriate metadata, it's going to be a struggle to find what you need, and often it can be almost impossible. If you're able to find what you're looking for, will there be enough information associated with that data and about the experiment to make it useful to you or anyone else? I like this cartoon with the woman looking at the wall of unmarked binders. She says, we should have labeled them. Yes. OMERO stores image data this way. Starting out with OMERO, you have a group name. Within the group, you have multiple projects, and you can have multiple data sets underneath each project, and you can have multiple individual pieces of data that are in each data set. Now that may seem like a shallow file structure if you're used to doing deeply nested folders, but OMERO is an object-oriented database, treating the data within these structures as objects. We can give the data additional attributes using tags and key value pairs and other annotations, allowing us to find specific data more easily. These annotations can be applied to projects at the project level, at the dataset level, and at the individual data level. To give you an example, this idea of tagging is something that Gmail uses pretty extensively. It allows Google to store just one copy of an email, but it can be tagged in multiple different ways so that it appears in different types of searches. And while it looks like folders, it's not. There are no folders. In fact, in Gmail, it's all search-based. If I give something the label of family in my Gmail account and I search for that, it'll show all my emails that I've tagged as family. If I was planning on coordinating a family cruise, I might find some of the same emails if I search for a different tag, something like 2025 cruise. To dig deeper, if I was looking for an email from my mom that gave me specific instructions about her diet requirements for the cruise, I could search for the tag 2025 cruise and then look for any emails from mom@gmail.com to create a small enough subset of emails so that I can find the information I need. Having key pieces of information will allow you to drill down to a much smaller amount of information from what could be tens of thousands of emails. This is taking advantage of object-oriented data rather than nested folders. If we look at the example on this slide, within this group we have a number of different projects, data sets, and individual pieces of data. This might not be how your lab would organize data, but we all know that over time stored data can get messy. If you're consistent in your use of tags and key value pairs, you'll be able to find relevant image data that might be located in multiple places. And you can even search for specific terms with more complex searches. If we had key value pairs for an organism that is a biopsy mouse, that might be this data. You might have cell lines that are HeLa. You might have treatments that are controls. Or you might have some data within that data set that are controls. You might have some H&E stains in certain locations, and you might be doing wound healing in others. But what if you wanted to look back at historical control images of your HeLa cultures because you're concerned that the culture may have become contaminated and you don't know when it happened? If you had well-annotated data, you would be able to make a more complex query. If you had well-annotated data, you would be able to make a more complex query and see your HeLa cells that are controls over a period of time. So how do we make our data more findable and useful? Tags are involved in the organization and categorization, and they can be used to substitute for the deep folder hierarchies that many labs are accustomed to using. Instead of thinking of file paths, you want to think in terms of data objects that will be accessible because of how they're annotated with tags. You want to use them in an intuitive, logical way, just as you might apply to nested folder hierarchies. Within OMERO, you can create tag sets. An example of a tag set might be microscopes, although this kind of data could also be a key value pair. Under a microscope tag set, you might have individual tags for the Zeiss LSM880 inverted confocal, the Leica DMI 6000, the Nikon biopipeline slide scanner, and so on. By having specific model information, which is likely buried somewhere in the instrument metadata, you can correctly state which instrument was used to collect the images. From a usability standpoint, we believe that every tag and tag set should include a definition of what that tag represents. The definition is to help group members understand what the tag means and how they should be applying it. It's a good idea to keep documentation on how your tags and tag sets are used. Unfortunately, tags and tag sets do not readily transfer between groups, nor is it possible to duplicate a collection of tags or tag sets to a different group, except of course doing it manually. Key value pairs are where you would categorize important information about the experiment, like the type of specimen, the sample preparation techniques, staining methods, genetic background, and all these different things that will help you understand the full scientific scope of the information contained in your image. You may want to organize your key value pair data by using community-developed metadata standards, such as REMBI, which stands for Recommended Metadata for Biological Images. There are links in the notes below. REMBI includes a useful checklist that your lab can use. Some labs that will be using OMERO will want to keep a separate structured vocabulary document open so that their key value pairs are used consistently. We strongly encourage labs to structure their data carefully and to standardize their terminology and spelling. OMERO is a database, and if things are not standardized, searches will miss data. For example, if someone in the lab is using the word renal and other people are using the word kidney, misspellings will also cause data to be missed. We've gotten spoiled with Google guessing what our search term typos mean, but most databases can't do that. Ontologies are another way to organize your data. If you've ever used PubMed, you've probably encountered the medical subject headings that are part of the structured vocabulary that the National Library of Medicine uses. Ontologies are structured vocabularies like MASH that have been carefully crafted by others, and they can be useful for organizing your data. An ontology has the potential to make your data more useful to others, particularly those outside of your lab. For example, if your NIH-funded data is shared after it's been published or when your funding has ended. If using someone else's ontology sounds like too much work, consider building a structured lab vocabulary for your lab. In my experience, most labs stick with a few standard cell lines or animal models. They look at the same tissue types. They often prep their samples for microscopy the same way. They look at particular biomarkers. They use similar stains as well as primary and secondary antibodies. This might be a time to have a lab meeting and come up with a file naming and annotation convention that you'll use consistently. Document the conventions and use that document as part of your onboarding for new lab members. Then, as needed, you can review the document to ensure compliance, as well as considering how it may need to be expanded. As you're thinking about your experimental metadata, consider how was the experiment performed, and maybe include reasons on why that methodology was chosen. You may want to document things like organism, gender, age, cell line, dishes, media, food, equipment used, and so on. If you're using cultured cells, when was the last time that the culture was tested to ensure that the cell line is correct, or it isn't cross-contaminated with other cell lines? You have the ability to add free text to specific data sets and projects as needed, but we encourage you to think about being very structured because, again, you can find better data if you use the same terms over and over again. References to specific lab notebooks and page numbers can be useful, and if you use electronic lab notebooks, hyperlinks are a possibility. And this is possibly important. Who did the experiment? Some labs have that person who maybe only lasted 6 months in their lab, and everything that they did didn't seem to work very well. Wouldn't it be good to know 3 years from now that certain data was processed and collected by that person? You might consider if you want to use that data, and if you trust it, that would be good information to know. The next question might be, what happened to your sample between the experiment and your imaging? Do you have standard operating procedures that can be referenced for your fixation, for your antibody staining? And if you used a service lab, do they have a versioned SOP that you can include? And you might include things about how the fixation was done as well as fixation time. Again, who prepped the sample? What organ were you looking at and what site within the organ? Who was the vendor and what was the catalog number of the antibodies that were used and the purchase date and maybe how long they've been in the freezer? And any stains that you used, primaries or secondaries, mounting media dishes, and so on. It's unfortunate that we as core managers see a lot of poorly prepared samples on the microscope. All of the university core facilities, whether you're doing microscopy, flow cytometry, mass spec, or genomics, we all want to help your lab get better research data. All you have to do is ask us for help. Technical metadata tells you how the image was captured. If you're using a computer-controlled microscope, like a confocal or even a core facility wide-field microscope, those instruments are going to capture a good deal of metadata about how the image was captured. In general, that data imports very nicely into OMERO. If you're using a less motorized microscope where you have to manually change the filters, adjust the rheostat to change the brightness, and all you do on the computer is choose your exposure time on a camera, that means you'll need to be more deliberate in capturing the technical metadata. We may be able to use a software tool called Micrometa app that was built by a lab at the University of Massachusetts. This app can in most cases be used to build what's called a JSON file, which is somewhat similar to XML, and it allows us to document your microscope's hardware. Once you have the file, then you'll still need to track the lens that was used, the filters that were manually moved into place, and all that. But the deeper technical information about the lenses and filters will already be available in the JSON file. Then OMERO reads that file, and the person adding the data can tell OMERO information about all their specific acquisition parameters. And again, who captured the images? Was it the person who uploaded them to OMERO or someone else? Using an available tool within OMERO called the iViewer, users are able to draw and annotate regions of interest on an image. In some cases, these ROIs can also be created from image analysis software that connects to OMERO using a plugin. The analysis software pulls down an image, performs a segmentation, and puts an ROI around the feature in the image. These externally created ROIs can be uploaded into OMERO by the analysis software and included with the analyzed image data. As an example, in this image, we've annotated this region of interest and called it a glomerulus. Again, using a structured vocabulary is highly recommended because if you plan to use AI to analyze your data, it's critical that the training sets contain well-annotated data so that they can serve as the ground truth, essentially to teach the AI what it is that you're looking for. OMERO does have the ability to do some data mining, which is basically a way to look for patterns and collections of information buried within larger amounts of data. You can find subsets based on various types of annotations like tags and key value pairs. Search might include tabular or region of interest data that's generated during image analysis. OMERO Parade is a plugin that we've added that allows you to look through all your data and come up with a subset that meets your criteria. There's a really good video about using OMERO Parade that came out of the August 2023 workshop that we held here at the U of A. We encourage you to look at that for more detail on finding subsets of data. The trainers were able to do things that I don't even know how to do. But if you're interested in learning how to find things in OMERO, the German bioimaging group has put together a bit of a treasure hunt using a public-facing OMERO group, so no login required, that allows you to practice using OMERO Parade, and I've included the URL here, and it's also available in the notes below. Here's some additional documentation about OMERO Parade, along with information about searching for data, annotating data, and filtering it. In addition to the workshop video that we mentioned, to email us for access if you're from the University of Arizona. There's also a nice video that's on YouTube directly that you can watch. I mentioned REMBI earlier. Again, that's recommended metadata for biological images. REMBI is a product of international collaborative effort to standardize scientific metadata. Consider that if we're our own most likely future collaborators, then this is the kind of data the future us will need to have to be able to reuse or even attempt to replicate the data. With all this sample prep and technical metadata that we've been talking about, do you still have enough information so that your experiment can be replicated? If you're not sure, you might need to collect more metadata. Remby has a very helpful template to remind all of us what's important data to collect. This slide gives you a quick idea of the scope of the Remby template. We have study component, the sample, more about the specimen that was collected, the image acquisition, the image data, how it correlates, how it was analyzed. It's just quite a bit here. I'd like to take a moment to talk about date formatting. Most of us have discovered as we work with people from other countries that are not part of the United States that there are different conventions with regard to dates. If we look at a computer file or a notation in a paper lab notebook that is 03-09-2024, is that March 9th or September 3rd? And there's a 6-month difference there, and that could be a significant problem. We encourage people to follow the ISO standards for dates. The standard formats date data in descending chronological order, which is year, 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 month, month, day, day. You can do it with or without the dashes. I personally like the dashes because I think it makes it more human readable. And remember to use leading zeros for months and days that are single digits. And if you're working with the files on a computer, you'll discover that they sort beautifully this way. I'd like to talk just a little bit more about data ownership. We've mentioned this before, but as a reminder, whoever uploads the data into an OMERO group is the data's owner. The group owner, along with some specific OMERO administrators, are the only other persons who can delete data that they didn't upload. And we encourage you to watch the OMERO groups video for more on this topic. One exception to this rule would be if you were set up in a read-write group. This permission level, which it requires an administrator to grant, allows anyone in the group to delete any data. Read-write groups assume a very high level of trust. If you as a group owner have a lab manager in your group or you work regularly with a core facility, there's the possibility of using a low-level administrative permission that allows specific people to upload data on someone else's behalf. This allows the designated person to become the owner of the data instead of the person who actually physically uploaded the data. Data ownership can be transferred to another user, but it's not simple. You have to have an administrator do that, and it must be done using the command line interface, which is basically Python. Since data ownership is tied to a specific user, the best practice is for a group owner to remove a user from their group rather than deleting the user completely from OMERO. And for group owners, remember that deleting a user can cause their owned data to be deleted as well. That's why you want to remove them from your group instead. This allows you to protect your group's data from loss, as well as from someone who you've decided should no longer have access to the group. And to protect data integrity, we created a generic common group in OMERO that everyone belongs to, since everyone has to belong to at least one group. Having that removed user sit in the common group gives the OMERO administrators time to transfer ownership of the data if that's what the group owner needs. Some other final things. Do you have a documented image analysis pipeline? There are some software packages that allow you to do scripting, and that gives your lab a way to be much more methodical with your image analysis. Are your annotations of your regions of interest consistent? This is where a dictionary document would be helpful to keep your nomenclature straight. Is your data AI ready? If you think you might want to use AI, your data needs to be consistently annotated and structured. AI can't do miracles with bad data. OMERO provides a number of options for organizing your data, and this video presentation, again from the German bioimaging group, is a nice introduction to the whole issues of data sets, key values, and tags. It's only about 20 minutes long, and the link lets you skip the beginning part, and the question and answer after the presentation is certainly very optional. To wrap things up, one of the legitimate complaints we've heard is that annotating data is time-consuming. There are ways that the OMERO user community has tried to ease this burden using add-ons, and there is an add-on that auto-suggests tags if you're doing nested folders. The add-on can read these and suggest tags to you. There are also people working on software tools that use Excel spreadsheets to add additional REMBE metadata that would be brought into OMERO as the image data is uploaded. But right now that's still in beta and it would require working with Python. We haven't done these yet, but if there's a lab that's willing to be a pioneer and help us incorporate or implement some of these, then what we learn working with your lab can possibly help other people. And please reach out to us using this project email address, which is also in the notes below. I want to take a moment to thank you. If you're watching this video, it's very likely that you watched the other two videos in this series. Thank you for putting up with listening to my voice for that long. Feel free to use this QR code to get our email address. Reach out to us. We're happy to help you get started, and thank you for watching.